Welcome to the Cell Series Endomembrane System video. At the end of this video, students should be able to describe the various components of the endomembrane system and their function. The endomembrane system is a set of organelle membranes that are intertwined throughout the cell. Some of the membranes are connected to one another directly, where other aspects are pieces of the membrane detached from the original organelles. The endomembrane system runs from the cell nuclear envelope through the cell to the cell membrane. Originating in the nuclear envelope, the endomembrane system continues to form through the rough endoplasmic reticulum, which leads into the smooth endoplasmic reticulum, passing vesicles through to the Golgi apparatus, where it leads to secretory vesicles and lysosomes, before finishing off at the cell membrane. The nucleus is often referred to as the brain of the cell. This is where all the genetic material for the organism lies. The genetic material, or DNA, is the blueprint for how to make an organism from conception through embryologic development and into adulthood, with all the necessary changes along the way. It is encased in the nuclear envelope. In this diagram, the nucleus is represented by number 1, which actually points to the outer membrane of the nucleus, but is referring to the organelle as a whole. Number 2 represents the nuclear lamina, which is made up of intermediate filaments, providing structure for the nucleus and holding the membrane proteins in place. The lamina also helps coordinate DNA replication and cell division. The nuclear lamina rests just under the double membrane of the nuclear envelope. Number 3 is pointing at the nucleoplasm, or nuclear matrix. As with the cell cytoplasm, the nucleus has a solution for holding in place the nucleolus and the chromosomes or chromatin, in addition to the amino acids, nucleic acids, and enzymes needed for nuclear enzymatic activity, as well as storing necessary molecules and ions. Number 4 is showing chromatin, specifically as euchromatin, which is the portion of DNA transcribed into mRNA. Number 5 shows the heterochromatin, which is the tightly coiled form of chromatin, containing inactive genes. Chromatin is more relaxed in general as compared to the DNA in its tightly coiled and condensed state as chromosomes. Number 6 references the nucleolus, which is a dense structure seen during interphase of the cell cycle. The nucleolus is where ribosomal RNA is made, and the RNA along with the proteins are assembled into large and small subunits of the ribosome, which is shipped out of the nucleus and used for DNA translation. Number 7 shows the nuclear envelope, more specifically the outer portion of the double membrane. The nuclear envelope is a highly sophisticated barrier that has to deconstruct and reconstruct itself during cell division. These deconstructed membranes may form the kinetochore, spindle fibers, and microtubules used in division, as well as centrosome interactions in the cytoskeleton. Number 8 is indicating the ribosomes. Ribosomes are the site of protein synthesis in cells and appear as tiny dots. Because the rough endoplasmic reticulum, which is indicated by number 10, is a continuous membrane with the nuclear envelope, both are studded with ribosomes. The proteins created from the nuclear ribosomes are transported into the perinuclear space between the membranes and the cell envelope, internal and external membrane. A side note, ribosomes are also found free-floating in the cytoplasm of a cell. Number 9 is pointing to the nuclear pore. A nuclear pore is a complex formed by nucleoporin proteins, creating an aqueous channel that allows bidirectional exchange of ribosomes, proteins, and RNA between the nucleus and the cell cytoplasm. Both inner and outer nuclear membranes are continuous with the rough endoplasmic reticulum, also called the rough ER or the RER. The rough ER double membranes form cisternae, which are similar to tubules, only flatter and thinner. These cisternae encase fluid, and the space that the fluid occupies is called the lumen. The rough ER lumen, or cisternal space, is continuous with direct access to the nuclear pores into the perinuclear space. The RER is called rough because of how the texture created by the studs on the membranes, which are ribosomes, looks under the microscope. The rough endoplasmic reticulum runs through the cytoplasm, and it is similar to a manufacturing plant. It synthesizes, packages, and assembles proteins for delivery to other organelles. The ribosome studs synthesize proteins, while the cisternae assemble the additional components the proteins need. Glycosylation occurs when the endoplasmic reticulum attaches carbohydrates to the proteins, creating glycoproteins. The proteins are collected in the cisternae to be folded until ready for packaging, to be sent off in vesicles to the Golgi apparatus for processing. Cell membrane proteins are also synthesized in the rough ER and added directly into the membrane. Ribosomes are responsible for synthesizing proteins in the cell. The ribosome compartments are made in the nucleolus and are composed of 4 strands of rRNA, or ribosomal RNA, and ribosomal proteins, forming 2 subunits. The larger unit on top is called the 60S, and the smaller unit on the bottom is called the 40S. When they exit the nuclear pore into the cytoplasm, they associate as one organelle, called the 80S. They don't associate until they are needed to perform a protein synthesis. The S is a value base settling weight when centrifuged. So that small pellet that forms at the bottom of a tube when it's spun very fast, that weight is 80S instead of, when you combine the 60 and 40, 100S. Ribosomes are not bound by a membrane and are much smaller than organelles. They are found in both prokaryotic and eukaryotic organisms. Ribosomes are found attached to the nuclear membrane and the rough endoplasmic reticulum. They can also be found free-floating or without being bound to anything in the cytoplasm. Ribosomes are also inside mitochondria and chloroplasts, although they are smaller, more akin to the sizing of prokaryotic ribosomes. Since DNA is too valuable to leave the nucleus, enzymes send out instructions to ribosomes using messenger RNA, or mRNA. The ribosomes use the genetic code translated into mRNA to synthesize the appropriate protein. Ribosomes also use transfer RNA, or tRNA, to bring the amino acids and attach them to the growing peptide chain. The rough endoplasmic reticulum merges into the smooth endoplasmic reticulum, also called the smooth ER, or the SER. The smooth ER is made up of a double membrane and cisternae. However, they are not flattened like the RER, but are more tube-shaped. They lack ribosomes, hence the smooth descriptor. The smooth ER has fluid throughout the lumen that holds lipids and steroids instead of proteins for processing. The smooth ER is responsible for making the lipids, including phospholipids, in the cell membrane, cholesterol, which is used in the membrane, as well as steroid hormones like estrogen and testosterone. An additional function of the smooth ER lumen is to hold calcium to regulate the uptake in the cytosol. Another function is to degrade toxins like alcohol. Glycosylation also happens in the SER, creating glycolipids, and after folding in the lumen, they're shipped off in vesicles to the Golgi, or to the cell membrane. The Golgi apparatus is like the post office. They receive, modify, package, and ship proteins, lipids, and molecules throughout the cell. The Golgi apparatus, also called the Golgi complex or the Golgi body, looks like a flattened stack of pancakes. The double membrane forms cisternae, and the interior fluid-filled areas are called the lumen. The lumen stores molecules, lipids, proteins, enzymes, carbohydrates, and other molecules for storage, modification, and sorting. There are 2 sides to the Golgi, the cis face and the trans face. The cis face is facing the cisterna of the smooth ER and accepts the package material after passing through each pancake or cisternal membrane for possible modifications. The material finally reaches the trans face, which is the face facing the cell membrane. It is transported out of the endomembrane system with transport or secretion vesicles into the cell for use or excretion. Transport vesicles with a special clathrin coating carrying cargo from the extracellular environment can also enter the Golgi apparatus at various enzyme-specific sites along the pathway. They are modified in reverse through the Golgi for shipping to the ER. Vesicles are transporters made of double membrane with cargo inside. Think of them like taxis or an Uber, transporting material throughout the cell, between the organelles, or to and from the membrane. Vesicles form by an invagination or pinching off of a piece of the double membrane of an existing organelle, like the ER or Golgi, for uptake or excretion by the cell membrane. Secretory vesicles are sent to the cell membrane for excretion. Other vesicles are used for storage, degradation, and protection. Lysosomes are the garbage disposal of the cell. They can be formed as vesicles from the Golgi and can take in transport vesicles from the cell membrane or uptake items from the cytoplasm. The lysosome breaks down or digests the material using hydrolase enzymes to denature or degrade particles. Hydrolases use hydrolysis to break covalent bonds. Typically, lysosomes maintain a low pH, under 5, as compared to the cytosol, which is around 7. Our cytosol is buffered to maintain the pH around 7 in case a lysosome ever breaks open. Lysosomes can degrade old cells, organelles, macromolecules, or cellular components that are no longer functioning properly. They can ingest or attempt to degrade pathogenic viruses and bacteria. They can also repair cell membrane components. Lysosomes are made of a single lipid bilayer with excess sugars, glycosidase proteins, integrated into prohibit the digestion of themselves and the cell. Vacuoles are like the pantry. They're used for storage of proteins, lipids, carbohydrates, or cell food, in addition to molecules or nutrients the cell might need. Some vacuoles store waste until such time as it can be secreted. Vacuoles are double membrane compartments that are larger than vesicles. Vacuoles in plants are larger than those in animals, and the central vacuole in a plant stores water. Some organisms regulate their cell-to-volume ratio using vacuoles, and others, like the paramecium, use a contractile vacuole to expel water to provide locomotion. Peroxisomes are a specialized lysosome that degrade toxic material. A byproduct of many toxic molecules is hydrogen peroxide. Peroxisomes degrade hydrogen peroxide, and they use catalase enzymes to hydrolyze 2H2O2 or hydrogen peroxide molecules, reducing them down to 2 water molecules and an oxygen molecule. They also metabolize amino acids and fats. Students should be able to describe the endomembrane system. Students should be able to compare and contrast the function of the SER, RER, and Golgi. Students should be able to differentiate between vesicles, vacuoles, lysosomes, and peroxisomes. Students should be able to describe the function and components of the nucleus of the cell.